G'day people, so I'm Shay and I'm part of the Rothnagel lab in Building 76, and my project's looking at the characterisation of peptides originating from upstream open reading frames. So what's an upstream open reading frame? Well, they're short open reading frames which are located within the 5' UTR of mRNA transcripts. Now the amount of UOFs you have in a transcript varies between transcript to transcript, but you normally have one about every 100 base pairs, so the smaller the 5' UTR, the less you have, the larger it is, the more you have on average. The length of UOFs is also variable, although the average is about 48 nucleotides, and where they're located within the 5' UTR is also not static, with some being located up near the 5' cap, some in the middle of the 5' UTR, and some overlapping with the main coding sequence. Now, UOFs can regulate the main coding sequence through several mechanisms such as ribosomal stalling and blocking, ribosomal reinitiation and leaky scanning. And all these mechanisms involve the translation of the upstream open reading frame. And our lab believes that these UOFs could therefore encode bioactive peptides which could have functional roles within the cell. So uPEPs are fairly hard to identify due to their short sizes, both bioinformatically and experimentally. In terms of bioinformatics, they're hard to identify because gene prediction programs look at the differences between coding sequences and non-coding sequences. And as UOFs are very short, they often lack the markers that coding sequences have. In addition, many gene prediction programs have a cutoff of 100 amino acids, so anything less than 100 amino acids is normally automatically classed as non-coding anyway. Experimentally, they're also hard to identify because of random mutagenesis. It's hard to, well, you're less likely to get a mutation within a UOF when compared to a larger sequence. Furthermore, for mass spectrometry, they normally have a cutoff of about 10 kilodaltons, and also when you're trying to extract the peptides from the cells, they're often subject to rapid degradation. Due to this, you have like an artificial discontinuity in databases, therefore, for identified proteins that are less than 100 amino acids, and that's what you can see in these graphs here. So this first graph here is the Phantom database, this one here is IPS database, this one's SwissProt. As you can see, below 100 amino acids, there's very few identified proteins. This last one here is the Phantom database again, but the authors used a different gene prediction program called Critica. They basically found 12% more UOFs, or predicted UOFs. So what evidence do we have for UOFs encoding bioactive peptides? Well, a number of bioinformatics studies have been performed on a variety of eukaryotes. First of all, in human and mouse, so Ikano et al., they identified 43 conserved UOFs. However, they found no evidence of protein coding potential. Crow et al., though, which is performed in our lab, they did identify 204 conserved UOFs, and they believe these did have protein coding potential based on higher conservation at the amino acid level, bias towards synonymous mutations, and about a third of the UOFs had optimal context for translation. The differences between these two papers can be attributed to the methodologies they used. So you've also had UOFs identified in Drosophila and other dipteran species, so you've got 44 conserved upstream open reading frames there, which are under mild to strong purifying selection and average about 70 amino acids in length. We've also had conserved UOFs identified in plants, so between Arabidopsis and rice, Hayden and Jorgensen identified 19 homology groups, all which are under strong purifying selection. Furthermore, 4 of these homology groups had conservation between other eukaryotes, including primitive plants, slime mold, fungus, and mammals. However, this conservation was for sequence and length, but they were in different mRNA transcripts. Therefore, the authors believe that these peptides will most likely be acting in trans. Tran et al. Also looked at plants. He looked at the 5 cereals, wheat, barley, maize, sorghum, and rice, and he identified 29 conserved UOFs for 5 out of 5 of those species. He identified a further 150 for UOFs that were conserved between 4 out of 5 of the species and 3 out of 5 of the species. He also identified 15 conserved UOFs between Arabidopsis and rice. Nisi et al. Looked at Cryptoctus neoformans, which is a fungus. He identified 122 conserved UOFs, but however, he didn't find any evidence for protein coding potential. Despite this, only 20 of his conserved UOFs are actually greater than 20 residues. Those 20 did have evidence of protein coding potential and also had a codon usage bias that was similar to other genes within the Cryptoctus genome. So this bioinformatic evidence indicates that UORFs are widely conserved throughout eukaryotes, both closely and distant related species. This conservation is seen at the amino acid level, and that there's evidence of purifying selection. However, bioinformatics doesn't mean that these UORFs are being translated. So you basically need further evidence, and this is sort of provided to us by Oyama et al., who used mass spectrometry to identify peptides originating from UORFs. And he identified 14 of these. And this diagram here shows 4 of the ones he identified. So the black boxes are the UORFs, and the white boxes are the main coding sequence. However, when our lab actually reviewed the 14 he identified, we found only 8 of these were actually coming from upstream uPEP reading frames. This can be attributed to basically databases being updated. So if UORFs do encode functional peptides, it would be expected if there was a mutation within the UORF that these could produce a disease. And Calvo et al. Performed a study and identified 509 genes where single nucleotide polymorphisms were actually creating or deleting upstream uPEP reading frames. And he identified another 8 mutations that were associated with the disease, and 3 were actually which have been shown to produce diseases. One of these, called Merino Uno-Hereditary Hypotrichosis, or MUHH, was further characterised by Wen et al. In 2009. They identified 13 mutations in the second upstream uPEP reading frame. You can see where the mutations are in this diagram here, all of which basically caused MUHH. An MUHH is a disease where you're born with sparse, wiry hair. As you grow older, this hair sort of falls out until you reach puberty, where you basically become bald. And this UORF was also highly conserved across 15 other mammals. So Wen et al. Also put the 5' UTR of the hairless gene into GFP and firefly luciferase constructs, and then deleted the upstream uPEP reading frame. And this produced increased expression of the main coding sequence. Due to this, they believe that this peptide was a cis-regulating element. However, I would argue that this isn't necessarily the case, because if you delete the upstream uPEP reading frame, you would expect to see increased expression of the main coding sequence, because you're not going to get ribosomal hallucination, leaky scanning, or ribosomal blocking or stalling happening, all of which actually do down-regulate the expression of the main coding sequence. So the hypothesis of my project is that UORFs do encode bioactive peptides, and this project will have, research into uPEPs has 2 significant elements. First of all, enhances our understanding of the proteome, and this also further helps us to understand how mutations within UORFs can best relate to disease. So the first aim of my project is to transfect HeLa cells with GFP uPEP fusion proteins. I'll be doing this by cloning my uPEPs into GFP C1 vectors, then transfecting these into HeLa cells, and then using confocal microscopy to determine their subcellular localization. Our postdoc Lexi has done work similar to this already in our lab, but she's been using GFP N1 vectors. So basically your peptides on the N terminus of the GFP, while mine's going to have the peptide on the C terminus, and we're doing this just to determine if you get different results. And these are the results we have seen with our GFP M1 vectors. And this is the U2HR subcellular localization. That was the gene I was talking about earlier. My second aim is pretty similar. However, I'll be transfecting HeLa cells with synthetic peptides, which we're getting produced commercially. These synthetic peptides will have a fluorophore end of them called FIT-C. And again, we just transfect these in the cells and use confocal microscopy to determine their subcellular localization. My third aim is to determine if uPEPs will produce differential gene expression by using a microarray. So I'll do this by transfecting one cell line with uPEPs and one cell line without uPEPs. I'll then extract the RNA from those cells, send the RNA samples to the Australian Genomic Research Facility, where they'll hybridise them to the microarray, do the image analysis, some basic bioinformatic analysis, then give the results back to me 5 to 7 weeks later. Whereupon I'll do more bioinformatic analysis to determine if there's any genes of interest, which will then validate using real-time PCR. So in summary, a subset of UORFs do potentially encode bioactive peptides. My project will aim to increase our knowledge about the role of uPEPs within the cell. I'd just like to thank Joe Rothnagle, my supervisor, and Lexi Friend, our postdoc, for providing a lot of help for me, and also other members in the Smith-Rothnagle lab. Any questions?